The next speaker is Maggie Aasen. Maggie is a biology major in the class of 2015. She's been doing biological research since her freshman year at Boston College and has been involved in a number of projects, from dissecting leeches all the way to creating stem cells. Besides science, Maggie has a passion for social justice and is involved in the Arupe International Immersion and Faith, Peace, and Justice minor programs here at BC. Maggie is writing her senior thesis on the health and environmental effects of current United States agriculture policies and subsidies. Let's give it up for Maggie.
Thanks for the introduction. And so as John mentioned, I work with induced pluripotent stem cells. But I actually first heard about these induced pluripotent stem cells years ago when I was a senior in high school in my AP biology class. And the way that my professor or teacher introduced me to these cells was by singing this song, anything you can do, I can do better. I can do anything better than you. And I'll spare you the performance because I'm not exactly musically inclined. But the point is that years later, I still remember that moment. And it's because after working with these cells for a year, I realized that they really can act as really better models for the future of medicine. And better models than the current type of stem cell or the traditional type of stem cell, which you've probably all heard of, which is embryonic stem cells. So I hope to, throughout these next 15 minutes, talk to you about my research and how my cells, iPS cells, can work as better models than embryonic stem cells. So you probably heard the term stem cells before, but I understand that not everyone here has been forced to take over 40 credits of biology these past 4 years as a bio major, so I'm going to try to catch everyone up to speed on some basics of cell biology. So you probably all know that we're all made of cells, about 37 trillion cells. But the thing about these cells is that they're not all the same. They're different, and they have different jobs. For example, we have our heart cells, or cardiac cells, and those expand and contract to push blood throughout our entire body. We have our liver cells, the cells that detoxify our entire body so we're not under this constant physiological stress. And then we have our skin cells, which you can obviously see on me and all over yourselves. And these protect our organs and keep everything kind of in place. So now you're probably wondering, well, this is talk about stem cells. So what do stem cells do? Well, that's just the thing. Stem cells don't have jobs yet. They're like many of you freshmen out there that haven't chosen a major, or many of you seniors out there that haven't secured a job yet. But you're all like freaking out. But the thing is, like our parents told us when we were little, we have the possibility and the potential to become anything we want to be when we grow up. And this is actually very true to stem cells as well. You see, stem cells have this ability it's called pluripotency. And this means that they have the ability to become any type of cell in the body. So all the cells that I just showed you, those heart cells, those liver cells, those skin cells, those actually once were stem cells. So now you're probably wondering, well, where do stem cells come from? Well, actually, we were all once a bundle of stem cells. We were embryos. And this is where the term embryonic stem cell research comes from. And it was actually started in 1998 when these couples started donating their fertilized eggs to researchers, and they were able to isolate these embryonic stem cells and start doing research on them. So you're probably wondering, well, why would I want to research stem cells in the first place? Well, the thing is they make excellent forms of therapy. And when I say therapy, I mean excellent replacement for damaged cells. So this is a picture of the Mona Lisa, obviously. Hopefully you guys know that. And on the left is how you and I would probably see the Mona Lisa. But on the right is how someone with macular degeneration would see the Mona Lisa. And macular degeneration is just this disease of the eye, and it causes this slow and really horrible vision loss over time. And it can affect anyone at any age at any time. So there is no cure for this disease, but in the past 5 years, researchers were able to implement these embryonic stem cells into people's eyes so that they would replace the damaged cells with new healthy ones, and so that people could see the Mona Lisa as it was meant to be seen. So not only do these cells make excellent replacement for individually damaged cells, but we can potentially replace entire organs. So right now, there are 123,000 people on the waiting list for an organ transplant. That's 123,000 people in need of a new heart or liver or kidney. And many people will wait for weeks to months to years for these new organs, and many people actually die waiting on this list. But with embryonic stem cells, we can completely bypass that problem. Essentially, we can create these organs in a petri dish so that people don't have to die waiting on that list. So obviously, this technology was at the turn of the century, and all these people are getting really excited about it. But with every new innovation, there comes some sort of controversy, and this is what happened in 2001 with the Bush administration. They started to limit the funding on this research because, as I just mentioned a few slides ago, it has to do with sacrificing human embryos. And so obviously, this was a large controversy, and all these researchers started to try to find new ways of developing these stem cells. And about 8 years later, they're about to do just this, and this is where induced pluripotent stem cells come in. You see, these researchers in Japan discovered that there's really only 4 major genes that cause a stem cell to act like a stem cell, and these are called pluripotency genes. So what these researchers did is that they isolated them outside of an embryonic stem cell and then inserted them into the genome or DNA of an adult cell, like a skin cell. And by doing this, they were able to essentially trick these cells into behaving like an embryonic stem cell. So now we can create stem cells out of skin, which is pretty cool, right? So obviously this is really amazing, and it actually won these researchers the Nobel Peace Prize just a year later, not only because it completely bypassed the ethical controversy of sacrificing human embryos for the sake of stem cell research, but it was also because these induced pluripotent stem cells, or IPS cells as they're called, they also have a unique property in that they're patient-specific. So that means that you can take your own skin cells and make them into stem cells. So one might ask, well, why is this really important? Well, let's think back to those 123,000 people on the waiting list for an organ transplant. Even after those weeks to months to years of waiting, even if we were to make organs out of embryonic stem cells, there's still a chance that after that time, these organs could be rejected. And this is because when foreign objects are inserted to the body, our immune system doesn't recognize them, and it actually attacks them. This is called organ rejection. But with IPS cell technology, we can completely bypass that problem. We can create organs made from our own cells. So therefore, our immune system will recognize it. It'll be like a puzzle fitting together. It's pretty cool. And that's why my lab started to get involved in this type of technology just about a year ago. But my lab actually doesn't use these cells to make new organs or for different therapies. We use them in a different way. So I kind of wanted to show you guys what I do. So this is a video of beating cardiomyocytes, or heart tissue, that were created in my lab this past January. So basically, it's not a full-on heart. It's just like a piece of a heart in a dish, kind of just sitting there. And so these were made from a stem cell line that I created about a year ago from mouse skin cells. And so these cells, they were never going to be transplanted back into a mouse, like say a mouse has a heart condition or anything like that. It's never going to be used as a form of therapy. We're actually developing this type of technology to use them as models. And when I say models, I mean models to model different genetic diseases and to develop different drugs. So you're probably wondering, well, how can I develop different drugs using this beating heart? Well, let's take a look today at the drug industry. And if you look at this graph, you can see that it's a really long and expensive process to create a drug. On average, it takes 10 to 15 years to create a drug, and in that time, $4 billion will be spent. And then less than 1% of these drugs will actually become approved by the FDA. So why does this take so long? Well, there's all these different trials of testing this drug. So it has to go through animal testing, and it has to go through rounds and rounds of clinical testing. And even after all this time, even if a drug does make it to that less than 1%, there's a chance that it might not even work for you. So why might not it work for you? Well, it's because we're not these cookie-cutter humans, you know? I'm different than every single person in this room, and everyone's different than every single person in this room. You know, we're going to respond to drugs differently. So I'm sure you've seen, you know, those commercials before, the ones with the magical butterfly that goes around granting, you know, all these magical hours of sleep to people with a flap of its wings. And then way at the end of the commercial, or way down in the fine print over there, in the size 3 font, they mention all the various side effects that they are forced to tell you about but really don't want you to hear. Well, the thing is, these side effects are a bigger deal than you think. About 700,000 people are sent to the ER due to adverse drug reactions every year. And 106,000 of those people will actually die due to these adverse drug reactions. That's 106,000 people that will die due to drugs that were prescribed to help them. That's twice the number of deaths from the deaths that happen in the US due to firearms and traffic accidents combined. So how can I test that a drug is going to affect me in a negative way before I take it, It's not like I can see into the future. It's not like I can clone myself and have the clone take the drug before I do and see how it affects them. Well, actually, we can do that with induced pluripotent stem cells. You see, I can take a piece of my skin, and I can make that into a stem cell. And then I can make it into any type of tissue that I want to test. So I can see how a drug is going to affect my brain, my liver, my kidneys before I take it. So this is this idea of personalized medicine. And it's really important because we all are different, I mean, if we all broke our legs here in the audience and we went to the emergency room, they wouldn't give us one-size cast that would fit it perfectly to our leg. And so this is why these type of cells are so important. And then also let's talk about that drug process. You know how it's so lengthy? Well, these cells are going to expedite the process of making those drugs. We don't have to have these long clinical trials anymore. We don't have to test these on animals, on rounds and rounds of human testing. We can essentially run clinical trials in a petri dish. So obviously you can see that this type of technology is really innovative. And not only can you use iPS cells to develop different drugs, but you can also use them to model different diseases. And this is what my lab specifically at BC is trying to get involved in. And when I say the word disease model, I mean a way to look at a type of disease outside of the body. So why would we want to really study disease outside the body? Well, let's see. Well, there's actually over 6,000 different identified genetic disorders for humans, and these are just a few of them that I've put up here, but there's 6,000 of them. And right now, there's really only 500 medications that are out there to combat these 6,000 diseases. And this is because the process for developing medications against these diseases requires extensive disease modeling. And the current disease models, I would argue, are less effective than they could be. It's a really timely process to create these different disease models. It's because we use mice. So basically what we can do with mice is that we can genetically engineer these mice to have any different disease that we really want. So like autism, Asperger's, cancer. And as long as we know the basics of this disease. But what about a disease that's rarer? What about a disease called progeria? It's a disease that affects children, and it's this rapid aging disease, and only about 250 children in the world have this disease. It's kind of hard to develop a mouse model of a disease that you don't know too much about. And there's another problem that's staring us right in the face here, and it's that we're not mice, and we're going to respond to drugs differently than mice. And I mean, a disease in a mouse is going to be very different than a disease in a human. And obviously, we can't throw a human into a cage and run a bunch of experiments on them. I'm pretty sure that's an entirely different ethical problem in and of itself. So this is where iPS cells can come in again. We can essentially create models of diseases in a petri dish. So what we can do is we can take skin cells from someone with a certain genetic disease, say autism, and we can turn those into stem cells. And we can look at the different tissues of a person with autism and see how the disease progresses throughout a person with a certain disease. And this also solves the problem of developing models with people with rarer diseases. We can take skin cells of the person of rare disease and create a perfect disease model of that specific genetic disease. So you can see that this type of technology is really incredible, and scientists are actually using this type of technology to study various diseases already. It's something that's being done today, right now. And it's the type of technology that my lab at BC is trying to get involved in. You see, we're still in the beginning stages of mastering iPS cell technology. We've already learned how to create iPS cells from skin cells, and now we're starting to master the different processes of differentiation, like the heart cells that you saw a few slides back. But once we do this, once we master these different processes, we hope to collaborate with labs all across Boston so that they can send us tissue samples from their patients, and we can create these genetic disease models and send them back to us so we can help expedite the process for finding cures and therapies for these diseases. So, I mean, I've really only touched the tip of the iceberg with the possibilities that this research has to offer. And I wish that I had another hour, but I really only have 30 more seconds. So I just wanted to say that this type of research is important. It's important for me, and it's important for you, and it's important for every person in this room. At some point in our lives, we become patients, or the people that we love become patients. And this type of technology is going to personalize medicine to make sure that we obtain the best treatment possible. It's going to help expedite the process to making these diseases, to finding cures for these diseases. It's going to make sure that it's certainly going to make sure that a medication that was given to you won't accidentally kill you. But really, if you get anything from my talk, I hope it's that really just through a small piece of your own skin, we can change the way that medicine works forever. Thank you very much.